So, welcome to your microbiology. Just so you know, I looked at your midterm for both this one and theory. So, I already have your kind of study guide already ready. I will be sending you guys that. It'll be on announcements. So I kind of wanted to put this on there so you'll be looking for it. I will go ahead and have it automatically populate for Monday, but just keep in mind we still have week 4 stuff to do, but I wanted you guys to have that so that you can have a little bit of a it's more like just security to know, okay, I'm on the right path, but I did look at a couple different versions because we know that they pull the questions. So I was able to get a good grasp, and you will receive those. It'll populate on announcements on Monday. Okay. Now, we have 3 chapters. However, chapter 11 is your mini session and it's already up on the research area. I'm sending you a link to that just in case you have not had the opportunity to look at it. This is the link to the mini session, just so that we're not having so much compressed in our live session. Today, we are going to break down chapters 8 and chapter 10. If we are able to get to our group discussion, we will. It's more how it affects us in the operating room. We do have a Kahoot, and then we're going to wrap everything up. Chapter 8, we're looking at the empire of viruses. So I did take some different things. I got 2 videos that we're going to watch. They're short and a lot of imagery to kind of bring it all together. We look at basic virology, tiny particles of nucleic acids. Viruses are so small. They're made up of basic genetic materials called nucleic acids. They're either DNA or RNA. These are the instructions for how they're going to actually function. They're surrounded by protective capsules. I want you guys to think of those as like the helmets that football players wear. So they have this protective barrier, okay? They're called capsids. These are that protective outer layer that keeps the nucleic acid safe, okay? Just like football players, when they're playing, they want to keep their head safe. So think of these capsids as that helmet. They cannot replicate on their own. Unlike bacteria and human cells, viruses can't make copies of themselves without help. They have to come into a living cell to actually do this, which is using the cell's machinery to actually multiply. Now, what you see in the slide on figure 8.1 just shows the relative size of viruses, of bacteria, and human cells to kind of give you that visualization of how tiny viruses actually are. Now, we look at how infections can be transmitted. Of course, inhalation and respiratory droplets. Big in the operating room. We have our masks on. When an infected person coughs, sneezes, or even talks, tiny droplets containing germs are just released into the air. If someone breathes into these droplets, they go where? Right into your airway, into your lungs. We look at the exchange of bodily fluids. Infections can spread when bodily fluids like blood, which we deal with all the time in the operating room, saliva, if you're doing a lot of oral maxillofacial cases, you're all up in the mouth, or other secretions from an infected person when they come in contact with a person's body, often through cuts, mucous membranes, or, of course, sexual contact. Eating, ingesting food, water, consuming contaminated food, we know can lead to illness. This is why proper food handling and clean water is extremely important. And then we look at bites by arthropod vectors. Some insects, mosquitoes, ticks, fleas, they carry germs. Now, when these insects bite a person, they can, of course, pass the germs into the bloodstream, which causes the infection. We get lysogenic and lytic cycles. This occurs when a virus hides its genetic material inside a host cell and then it just stays inactive, or we call it latent. As the host cells divides and creates new cells, each new cell receives a copy of that viral genetic nasty material. This means the virus can spread quietly, silently, kind of like that hidden person in the back, okay? Without activity causing an infection until it's triggered to become active again. One of the biggest things that I think of is rabies, right? You get rabies, and oftentimes when it's fatal is at the latent stages, okay? We get bacteriophages, viruses that invade bacteria. Crazy, right? Got viruses that are actually invading bacteria. These viruses, or you can say phages, they specifically target and infect bacterial cells. So what they do is they bind to receptor sites on the bacterial host cell wall, like the flagella, like the little tails, all right? They attach themselves to specific sites on the surface of bacteria, and then they are like docking at certain structures, like they're just staying in place of that cell wall. They have a more, I would say, complicated structure compared to other types of viruses. So they're referred to as a complex virus. You may see that again. These are complex viruses. They're classified as either virulent or temperature. Virulent phages immediately take over. They go in and they're destroying the bacterial cell to reproduce. Temperature phages, they can remain dormant within the bacterial cell and replicate along with it until there's like a trigger, and then they become active. We also have harnessing bacteriophages. I want you to think about antibiotic resistant and phage enzymes. We start off with antibiotic resistance. Antibiotic resistance has begun to increase at an alarming rate. And that's because, unfortunately, bacteria are becoming stronger and more resistant to antibiotics, making it harder for them to treat infections, which is important for us because in the operating room, we do not want a patient to get one of these antibiotic resistant um, viruses, bacteria. This is growing exponentially in medicine. Scientists are developing and experimenting with different enzymes that phages produce lysis with their hosts. So, what they're doing is they're studying special enzymes looking at viruses that infect bacteria. Now, these enzymes help kind of break things down and kill the bacteria that they infect. They're also exploring whether enzymes could be used as an alternative treatment to fight these antibacterial resistant bacteria. Because, we give out a lot of antibiotics. In the operating room, we do put antibiotic items in our fluids. Sometimes anesthesia does it. Now, with latent viral infections, some viruses can stay hidden again in the body for a lifetime without causing any symptoms. They only become active and cause disease when the conditions are like just right. Like when the immune system is weak, they have pneumonia, they're, just under the weather or stress. This ability to remain dormant and then re-emerge oftentimes makes them tricky to kind of just deal with in the first place. We do have antiviral vaccinations. Now, one thing to keep in mind is antibiotics have no effect on a virus. Antibiotics are designed to kill bacteria, not viruses. So, taking antibiotics, it's not going to help you if you have a viral infection like the flu. Antiviral vaccinations, vaccines that are developed to help the body fight specific viruses, will. They prepare your immune system to recognize and attack a virus if you actually get exposed to it. I want you to think of like smallpox. On the ship, everyone had to get the smallpox vaccine um, just to help prevent if we came into an area where we were exposed to smallpox, our bodies would have built up some type of immunity towards the smallpox disease. Now, immunizations cause the body to produce antibodies. When you get vaccinated, your body creates those antibodies that are going to target that particular virus, i.e., the smallpox. If the virus tries to infect you later, these antibodies usually come in quickly and they respond to stop it from reproducing, getting you sick, and causing illnesses. We look at interferons and glycoproteins. Interferons are special proteins with sugar molecules attached to them. Their main job is just to come in and be that superhero to fight off viruses. What they do is they come in and interrupt the replication of the RNA and DNA viruses. Interferons work by interfering with the ability of the virus to make copies of themselves, which is good because then it slows down or stops the spread of virus altogether. We have two types. We have leukocytes, which produce one type of interferon, and leukocytes are white blood cells that help fight these infections. We also have lymphocytes. Lymphocytes produce another type of interferon. Lymphocytes are specific groups of white blood cells involved in the immune response. You have lymph nodes all over your body, okay? But you would think of them if you guys take a deeper microbiology course, you'd start talking about T cells and B cells. All right. We're going to get into common viruses. We get into herpes virus. Okay. We have herpes simplex virus, and we're going to go in more detail with this particular virus later. But here's just some breakdown of some of them. They cause cold sores. You have HSV1 and then genital herpes, which is HSV2. We also have VZV, varicella zoster virus. This virus causes chicken pox and can reactivate later in life. And you hear a lot of older people now getting the shingle vaccine because later in life it's shingles. We also have Epstein-Barr virus, EBV. This virus is best known for causing mono, or as they say, the kissing disease. Human herpes virus 6 and 7. These viruses usually cause mild childhood illnesses such as rosella. We also have gastrointestinal viruses, the rotavirus, which is a common cause of severe diarrhea in infants and young children. Norovirus, this one is the stomach flu. Okay, it's highly contagious and causes vomiting and diarrhea. And we also have astrovirus. These viruses can also cause stomach issues like diarrhea, but particularly in children and the elderly. Enteroviruses. Now, these viruses mainly affect the gastrointestinal tract, but they can spread to other parts of the body. They're quite common and cause a wide range of illnesses. Another one is the rhinovirus, the most common cause of the common cold. You may see this somewhere again. It primarily infects the nose and your throat. Polo, and this is where you see this picture on your screen. Now, polovirus, this is a serious illness that can lead to paralysis. Vaccines have helped control it, and that's where you can kind of see a lot more in 812, which is a 1960 photograph of a nurse carrying a victim. The last individual who was in the iron lung, I believe he had just passed away, but he was in that I think he got it when he was a kid, and up until old age, he lived in the iron lung. We have Coxsackie virus. This can cause hand, foot, and mouth disease, which leads to a rash and sores, of course, in these areas. It can cause it, when you think of it, it's more like a mild cold-like symptoms. But there are more serious complications in rare cases, of course, because it's not just a cold, right? It's hand, foot, and mouth. Another one that looks like the cold is ecovirus. Symptoms would be fevers, rashes. It can sometimes lead to more serious infections like meningitis. And then we also have the non-polovirus. This refers to another enterovirus that aren't polovirus, but can still cause illness ranging from just mild respiratory infections to more severe infections of the brain and the heart. Now, in the operating room, we treat every patient as if they have everything. So we mask up, we have our gloves on, our gown, our eye protection. We look at respiratory viruses. These viruses primarily affect the respiratory system and can cause illness ranging from a mild cold to severe respiratory infections. RSV, this is respiratory syncytial virus, a common virus, of course, that causes respiratory infections in infants and young children, leading to bronchiolitis and pneumonia in severe cases. Coronavirus. The family includes several viruses that cause respiratory illness, ranging from the common cold to more severe diseases like COVID-19. And then we look at influenza A, which is a major cause of seasonal flu, and it can lead to widespread outbreaks and can affect people of all ages. But it's important to get vaccinated to help, of course, prevent its spread. Now, I mentioned hepatitis or herpes, but we're going to get into hepatitis, okay? Now, these viruses primarily affect the liver and can cause inflammation leading to various liver disease. The one thing that I want you to know is that you're going to want to know how to differentiate the versions of hepatitis from hep A all the way to hep E. Okay, so let's try to break these down. You have the chart, and I'm going to try to give you guys this slide with my notes on it so you guys can have it. Hep A, transmitted mainly through contaminated food and water. Okay, Hep A, food and water, causes acute liver disease, but does not lead to chronic infection. A vaccine is available for prevention. Hep B, HBV, spread through contact with infected bodily fluids like blood or sexual contact. It can cause both acute and chronic liver infections, leading to serious complications like cirrhosis and liver cancer. The vaccination is effective in preventing infection. Okay, so you got Hep A, Hep B, Hep C, mainly transmitted through blood. And what I mean by blood, sharing of needles. It often leads to a chronic liver infection, which can cause long-term liver damage. There is no vaccine. I repeat, there is no vaccine for Hep C, but there are antiviral treatments can effectively cure the infection. Okay, so we got Hep A, Hep B, Hep C. When we get to HDV, hepatitis D, this virus can only infect those already infected with HBV, which is hepatitis B. It makes hepatitis B infections more severe. Vaccination against HBV can prevent HDV, okay? But keep in mind, you can only get HDV if you already have HBV. And then the last one on our chart you see is HEV, hepatitis E, transmitted primarily through contaminated water. Again, this causes acute liver disease, especially in areas with poor sanitation. A vaccine is available in some countries, but is not widely used. All right. So you're going to want to make sure that you can identify these if for some reason you see them. We get to HIV, human immune deficiency virus. This is a virus that leads to acquired immune deficiency syndrome or AIDS. HIV attacks the body immune system, specifically the CD4 cells. Remember, I was telling you about those T cells if you want to get deeper into micro. And these are important for fighting infection. So it kind of breaks down your immune system. If untreated, HIV can weaken the immune system over time, making it difficult for the body to defend itself against other infections and diseases. Now, there is no cure for HIV, but there are ARTs, which are antiviral therapy that can effectively manage the virus, allowing individuals to live a lot longer than they were when the disease first kind of came on the scene, where now they're living healthier lives and significantly reducing the transmission of the virus. Now, viroids. Viroids cause disease in food crops. Viroids are tiny infectious agents made up of a small circle of RNA. They can infect plants and lead to diseases that affect crops, health, and how much they can yield. Only recognized defense is a complete destruction of the entire crop. So as soon as a viroid is on that crop, they have to destroy the entire crop. And that is the current only effective way to prevent the spread of these viroids to other plants. Now, this helps to stop the viroid from spreading to healthy plants as there are no specific treatment available to cure the infected crops. But I wanted to kind of show you a quick video from the science show that talks just about viroids. So let's take a look.
In 1971, a USDA plant scientist named Theodore Otto Diener discovered what could be the smallest pathogen on the planet. It was unlike anything biologists had ever seen. It could make copies of itself like a virus, but was much, much simpler. It didn't have any kind of outer protein coating and just a tiny bit of RNA. At the time, scientists didn't think something like this could even exist, but these pathogens called viroids were very real. They infected potatoes, turning them into gnarly twists that looked more like ginger than something you'd top with sour cream. And researchers eventually realized they might be a clue to how life got started on Earth. As far as plant diseases go, potato spindle tuber disease wasn't a life or death situation like the Irish potato famine in the mid-1800s, but it was still a problem for farmers and no one knew what caused it, so the USDA was put on the case. Theodore Diener and his colleagues figured the potato disease was caused by a virus, but when they extracted samples of it, they discovered it was much smaller than any known virus. It was subviral. And that was baffling. Tests revealed that, unlike viruses, this thing didn't have a protein code. It was just a chunk of RNA, or ribonucleic acid, the molecule we usually think of as a template used to make proteins. At the time, scientists had also assumed that even small pathogens needed a certain amount of DNA or RNA to replicate themselves, at least a couple thousand nucleotides. That's tiny compared to most living things, but enough instructions for a virus to take over a host cell and make copies. But this potato menace, whatever it was, seemed to have just a tenth of that. Because Diener's mystery agent was similar to, but also clearly different from true viruses, he called it a viroid. We now know that viroids are tiny, single-stranded circles of RNA. Unlike RNA viruses, the RNA isn't used to make proteins. Instead, viroids make their way into a plant cell's nucleus or its chloroplasts, where photosynthesis happens. There, the viroid cranks out copies of itself using the host's RNA polymerase, the enzyme that strings together pieces of RNA based on a DNA sequence. Some viroids also have the ability to act as ribozymes and cut RNA, which helps their replication process. Today, we know about dozens of viroids, which infect lots of crops beside potatoes. There's the avocado sunblotch viroid, the apple scar skin viroid, and the hop stunt viroid. Some viroids cause minor cosmetic damage, but others, like a viroid that infects coconuts, will kill the entire plant. They're also sneaky little devils. Different viroids can get into plants in a lot of different ways. Farmers need to be on the lookout for contaminated farm tools, seeds, pollen, and even insects. Aphids, for instance, can suck up millions of copies of viroids when feeding on an infected plant, which they can pass on to an unsuspecting victim with their next snack. Fortunately, viroids don't infect humans. The closest thing is hepatitis D, which looks an awful lot like a viroid, but technically isn't, since its RNA codes for a protein and it doesn't self-replicate. But even if we don't need to worry about viroids giving us sunblotch or a scarred skin like a poor infected apple, they're still important to study. They could even give us insight into where we and all life came from. That's because some scientists see viroids as living fossils, precursors to all of the DNA and protein molecules in organisms today. RNA is a unique molecule. It can be passed on between generations to preserve information, and it can sometimes do things, like catalyze chemical reactions. Those are two things that cells really need. Many biologists support an idea known as the RNA world hypothesis, which suggests that life on Earth began with RNA, and later, evolution drove the creation of the
All right, they were getting ready to go on a whole nother tangent, so I wanted to make sure we stayed on point with the viroid. But you can kind of see how the viroid different types can get into different plants, crops. Okay. The last part of chapter 8 that we're going to talk about is the prion. Okay. Now, these are a group of rare, progressive, neurodegenerative diseases caused by abnormal proteins called prions. They lead to brain damage that creates sponge-like holes in brain tissue, resulting in severe neurological symptoms. Um, you have GSS, which is Gertzmann-Straussler-Scheinker syndrome. Say that 1,000 times, which is a rare inherited form. It's typically caused symptoms like difficult walking, loss of like coordination, cognitive decline, usually beginning in middle adulthood. You have FFI, fatal familial insomnia, another inherited disease. Individuals with this experience progressive insomnia, leading to severe sleep deprivation. They are usually confused and ultimately, unfortunately, death, often within a few months to a few years after the symptoms begin. Another one is Kuru, Kuru. Now, this one was hysterically seen in certain tribes in Papua New Guinea associated with cannibalism, where they were actually eating the brains of deceased individuals. So, and symptoms here include tremors, loss of coordination, severe cognitive decline, and fortunately, it's rare due to them stopping the practice of cannibalism and eating brains. So there you go. Now we're getting ready to go into chapter 10. All right. I'm going to pose this question to everyone here. As a surgical technologist, how would you be able to identify and manage the risk of parasite transmissions in the operating room? And as you're thinking about this question, we're going to go through the chapter, but I want you to look and think about identifying different risks. Okay. Looking at managing those risks. And what I mean by identifying the risk, I want you to think about what in a patient's history assessment would help us to identify. What symptoms would we be looking for? When you start getting into your procedures, how important is it to be aware of what you're doing in the procedure? For example, let's say we're doing an abdominal surgery or those involving contaminated tissues. So now you got to think, what is in the intestines that could potentially cause problems if whatever was in actually gets out? With managing risks, I want you to think about infection control protocols, looking at PPE. Why is it so important to handle certain specimens? Think about the training. Okay. Again, time permitting, we will break into different groups and talk about this. But let's get into an overview of parasitology. Now, we look at invertebrates as human pathogens. These are animals without backbones, and some can cause disease in human parasites. And so that's why you see the little jiffy there. Okay. We have different types. We have the parasitic worm, we have roundworms, we have flatworms, we have ectoparasites, insects, fleas, lice, ticks. They can transmit other pathogens. And then they can enter through the body. Ingestion of contaminated food, contaminated water. Um, they can penetrate the skin, like the hookworm larva, bites from different vectors. So in table 10.1 that you see on the slide is just a list of common parasites, their transmission methods, and diseases that are related to them. All right. So let's talk about types of human parasites and still be thinking about that question as we get into this chapter. There are different types of protozoa. Okay, these include amboas, where they move temporarily like false feet. They don't have real feet, so it's false feet. We have amoebios, okay, right here. These ones would be which would cause dysentery. We have flagellitas. They have that whip-like tail that helps them with their movement. We have kind of bringing them all down, ciliates or ciliphoras. They have numerous hair structures like cilia, and that's how they get their movement. We have spore zones, generally non-modal. They're very slow, especially when they get into the adult form. And microsporidias. These are intracellular parasites and they have protective spores around them. Okay, so looking at protozoa, just know these are all types. All right, but we want to know about the parasites. We look at rickets, okay, rickettsia. These are a group of bacteria that are obligate or intracellular parasites, meaning that they have to live inside host cells. That's all that means. They have to live within. They're often transmitted by arthropod vectors like ticks, fleas, and mites. They have different groups. We have typhus group. These cause epidemic typhus. They are transmitted by lice or fleas. So lice would be an epidemic, fleas would be an endemic. The symptoms would be like high fever, headache, a rash. They may start to feel muscle pain. We also have scrub type or scrub typhus group. Now, here, these are transmitted by chigger mites. Again, fever, headache, rash, or you'll see like a dark sore or eschar. So eschar means dark sore, okay? Spotted fever group causes Rocky Mountain spotted fever, again, transmitted by ticks. Now, these symptoms appear about 2 weeks after the infection, and they include like an onset of a high fever. Person has like huge headache. They're really tired, muscle pain. They're going to complain of chills. They're going to have a rash that's going to begin at their extremities, and then it'll move to the palms of their hands and the soles, and then it'll go to the rest of their body, kind of like what you see in figure 10.5. Now, key points. It's often present with flu-like symptoms, okay? There's a specific rash, and depending on the group. So what you have to do is have prompt diagnosis, and treatment is going to be important because you don't want to have severe complications with this. But that's not the only human parasites. Chapter talks about metazoa, multicellular organisms that can be parasitic in humans. You have parasitic worms that infect us. Again, I mentioned roundworms, tapeworms. These are going to be organisms that live on the exterior of the host, like on our skin, right? Um, Common arthropods, fleas, ticks, lice, mosquitoes. Now, why is this medically significant? Well, arthropods can bite, sting, infest humans, but rarely secrete any type of harmful toxins. Their primary role is crazy because they're just disease carrying. They're just going, bloop, gonna grab disease here, bloop, gonna pop it on this person and keep it moving. So they're transmitting various infectious diseases. And so you see the different examples of these vectors in figure 10.7. Mentioning vectors. Okay, so they are just organisms that they're not going to cause disease themselves, right? But they're extremely important because they're the ones spreading the disease, bacteria, viruses, parasites from one host to another. Type of vectors. We have the chapter talks about biological vectors, organisms in which pathogens undergo development or they multiply before they actually transmit to the new host. So they have it in their body, they're making more of it, like more soldiers, and then they are able to dump a whole bunch of the vector or whatever in them into the new host. For example, mosquitoes, they spread malaria and dengue virus. Ticks, Lyme disease, Rocky Mountain, spotted fever, rickets. You have mechanical vectors. These are organisms that carry the pathogen on their body without any developmental change. So it's just on them. Okay. Fleas, for example, they can carry bacteria like Salmonella or E. coli on their bodies and then contaminate the food. So they're not biting into the food or anything. It's kind of like, okay, I'm going to just shake it off. Right. And then there it is. The role in disease transmission. Now, vectors, they play a critical role in the life cycle of many pathogens and are often important for their transmission to us as humans or to any other host, making them significant in why we study infectious disease. Now, there are vector-borne diseases. The definition of a vector-borne disease are any illnesses caused by pathogens that are transmitted from one host to another through vectors such as insects or arthropods. Vector-borne diseases can cause significant morbidity and mortality worldwide. For example, malaria, which is transmitted by mosquitoes, or Lyme disease transmitted by ticks. And so you hear me saying these diseases again because you may have to identify, hey, which disease could potentially cause what type, or which vector could potentially cause what type of disease. Okay. Now, we look at animal and plant health. These diseases also look and affect livestock and crops, leading to economic losses and food security issues. Researchers face difficulties in predicting vector behavior, which can be influenced by a lot of different factors like environment, the temperature, humidity, changes in land use, and urbanization. It's important for us to understand these vector behaviors because we're trying to effectively control measures and prevent strategies or create preventative strategies for vector-borne diseases. We do have host-parasite relationships. Types of parasites. We have the obligate parasite. These parasites require a host to survive and reproduce. They cannot live without or live outside of the host. We have opportunist parasites. These parasites typically, they're not going to cause disease in healthy hosts, but they can exploit weakened immune systems or other vulnerabilities to cause infection. When we look at infection with a parasite, that does not always lead to damage or disease in the host. Many factors influence the outcome of infection, including the type of parasite, the host's immune response, the presence of other conditions. Some parasites can actually coexist with their host without causing any type of harm at all, leading to a state of communalism, where one organism benefits while the other is neither helped nor harmed. You look at table 10.3 in the book, and it gives an overview of the microbial relationships, including examples of different types of parasites and their infections with their hosts. There is also what we call a survival mechanism. We look at asexual reproduction, where we're involving the production of offspring without the fusion of gametes. There's something called binary fission, where the organism divides into identical cells. There's also budding, where new individuals develop from a bud or an outgrowth from the parent. With sexual reproduction, it involves the fusion of a male and female gamete to produce an offspring, often leading to genetic diversity, which can enhance survival, especially with the way the environments change. We look at the life cycle of parasites. It involves multiple hosts at stages of development. Parasites may undergo different forms, larval, adult, and reproductive methods in a lot of different hosts. For example, we look at Plasmodium species, which causes malaria. It has a complex life cycle involving humans and mosquitoes. There's also simple life cycles. These typically occur in a single host where the parasite develops and reproduces. The life cycle is less complicated with fewer stages and transitions. Examples would be Entomobia histolica, which causes ambiotic dysentery, primarily exists in a single host or a human. As we get into a little bit more of malaria. Now, malaria is caused by Plasmodium parasites, which are transmitted through the bites of infected female mosquitoes. You might want to know that. We look at the primary pathway for malaria transmission is, of course, through the bite of a female infected mosquito. I'm going to say that again. The mosquito injects the parasites into the human's bloodstream. The blood transfusion from an infected donor, you can get it. Sharing needles contaminated with infected blood is another way you can get it. Mother to infant, so congenital transmission from an infected mother during pregnancy at childbirth. But there are populations that are more at risk. Young children, their immune systems are still developing, making them just that much more vulnerable. Pregnant women, pregnancy weakens our immune response and again increases our susceptibility. The older population, they may have reduced immunity, making them again more at risk for severe diseases. And then we look at the immunocompromised individuals. Now, these are going to be people that already have weakened immune systems due to conditions like HIV, AIDS, or other diseases that are at a higher risk for severe malaria. Now, malaria is the most common parasitic infection in humans worldwide, particularly prevalent in poor countries located in the tropics or subtropics. Why am I talking about that? Well, we look at neglected parasitic infections, and this is the last part of the chapter. MPIs are a group of parasitic and bacterial diseases that affect more than 1 billion people worldwide. They are often overlooked in terms of research and public health funding. Now, the impact of neglected parasite infections are health consequences. We look at impairing physical, cognitive development, particularly in our children. They contribute to illnesses and death in mothers and in their children. It increases susceptibility to other diseases and other conditions, but that's not just that. There are eco or socioeconomic effects where it makes it challenging for affected individuals to farm or earn a living, leading to economic instability. There's limit productivity in the workplace, resulting in a broader economic consequences for communities, for countries. We look at how they kind of come about. And I found this video by none other than Bill Gates. Now, before we jump into our cahoots, let's take a look at this video and see what he has to talk about these neglected tropical diseases.
In the tropics, you have a number of diseases that you don't see anywhere else, and they're often quite horrific. We have more than 1 billion people who are at risk of these diseases. There are things like worms that are in your intestine, taking away your nutrition, and that stops kids from being able to learn in school or parents from being able to work. Guinea worm you get when you drink water that's got these small worms in it, and then it grows inside your body. And as the worm gets larger and larger, it causes intense suffering, itching. So people typically go get in the water, which actually allows the worm to escape. It actually leaves your body and goes back in to create its life cycle. Well, elephantiasis, there's a set of worms that actually go and live inside your lymph nodes. Your lymph node just grows very dramatically. And so whenever you see very extreme things where somebody's leg is 10 times the size it should be, or other parts of the body, that's typically elephantiasis. River blindness causes you to go blind. If you live in a rural village, that's even worse. There's no braille books, farming type work that you would normally do. It's very tough. Amazingly, there are drugs to help treat most of these diseases. And so we worked with the drug companies to get huge donations and donors to fund getting out and having everybody who needs them take these pills. And if we do this well, a lot of these diseases can actually be completely eliminated.
All right. So you can kind of see, kind of makes sense. A lot of these places are farther out. They don't have the access. And so I thought it was really interesting that when I came across this to kind of share, hey, look, there are people that do care. With that, what questions do you have for me? All right. Well, I have a few questions for you all. If you are driving or somewhere where you are not able to log in, that is okay. I am loading the game. There are 8 questions. These questions you may see from your knowledge check. I did look at some concepts from this week's quiz. And I wanted to kind of throw them out at you. All right. Again, if we do have time, we will get to that question. But if we do not, that is okay. I want you to kind of save it for next time. So there are 13 of us. Let's see how many can log in. And again, if you are not able to log in, that is okay. Just send me a message later so that I will know. But if you're driving, don't send me the message while you're driving. Just send it to me later. I got you. All right. There are 8 of us on. I know 1 is driving. I'm going to wait a couple more seconds. Okay, 2 driving. All right, let's go ahead and start. Everyone else will be able to kind of watch the playback, okay? Again, these are questions that you may see this week. Again, these are questions that you may see. Question 7. Okay, last question, last question. I look like I put the question in one of them. All right. I have to fix that one. Good job. Good job. Good job, everybody. All right. I think the music is still playing. Hold on, let me turn that off. All right. For those of you who are watching the playback, what considerations should an ST consider regarding the impact of traveling outside of the United States on blood, tissue, or organ donation? How might this affect infection control and patient safety in the operating room? And you have the same opportunity to answer this question now that you guys will answer before we do. As a surgical tech, how would you identify and manage the risk of parasitic transmissions in the operating room? Today, we covered essential information about viruses, viroids, prions, parasites, and vectors. In Chapter 8, we defined key terms and explored how non-living viruses, viroids, and prions reproduce and spread. In Chapter 10, we focused on parasites, comparing various human parasites and their transmission methods. We discussed several mechanisms and highlighted the resurgent. We looked at those neglected diseases in the United States and abroad, and really just emphasized the need for awareness and prevention, and had Bill Gates kind of show us what he's doing. We are coming up on our midterm. I encourage you guys really to just stay engaged, continue to work like you guys are doing. You guys are doing a really good job. I know that a lot of information is coming at you very fast, and you guys are really doing good. Don't let anyone tell you otherwise. Don't geek yourself out. You guys are really doing good. If you have any questions or need any type of clarification, you guys, I am here. Please reach out. Let me know, especially as this midterm is coming up. As I mentioned, your midterm review will be coming out on Monday morning, just so you can have it. That way you don't have to stress, but still prepare for week 4, okay? I am wishing you guys all a wonderful evening and productive rest of your week. Stay safe this weekend, and I look forward to seeing everyone who can join us for next week's live session. Good night, everybody.